I'm just going to wait one more minute until I get a few more people jumping in. It's looking pretty close though. Do the TAs have any announcements or anything for the group, or should I just jump right in?
We just want to remind everyone that you need to check the Canvas announcement to sign up the presentation for the discussion section. And that's it.
Okay. So I'll get started. It looks like we have most people here. So good morning or early afternoon, everybody. I'm Jesse Reinhardt, and I'm going to have 2 lectures with you. So today and then 1 lecture next week. And just getting my clock here so I can stay on time. So the majority of our time together is going to be spent on proteomics. Okay, and so my role in this course is to introduce where, I mean, this Marco and I always talk about is like, where does the data come from, right? Because, I mean, as you well know, this is a data mining course. It's about gleaning omics data and trying to integrate that into computational workflows. But in order for you to do that, it's important to just get a really good sense of where the data comes from. So you've had probably crash courses in nucleic acid data discovery, DNA, RNA. And so you need a little bit of information about proteins. So my job is to cram maybe 2 courses of information into about 1.5 lectures. So I'm just going to warn you in advance, I have data-rich slides, but this will be a resource for you to take it or leave it. I'll explain most of the stuff on the slides, but there will be references, case studies, annotations to the literature that's there for you should you want to explore more, if you want to clarify something. We don't really have formal office hours for the different instructors in this course, but I'm more than happy to meet with you one-on-one if there's a critical component that I can't cover. Again, it's 1.5 lectures. We're going to get into what I think are every important area in this vast field. And then I'm also going to change gears radically, and I'm going to introduce structural biology, because you will be sampling structural information about proteins and possibly nucleic acids. And so I have a bit of a background in that. So I'm going to just give you a very broad strokes introduction to that. So as I talk today and you have some questions, feel free to jump in. We'll try to manage the questions and the discussion, try to keep it short, but I'm encouraging you to go ahead and ask questions as we go, okay? So I'd like to start with this big picture slide where we see what you should be very familiar with as a central dogma, DNA, RNA to proteins, that I just explained, that we're focusing on the protein aspect. And you know very well it's all interconnected. But these images that I always put here underneath the central dogma is to give you a sense that, again, you should already know this as students in biology, whether it's primary interest or even a peripheral interest of yours. We know that the information contained in these molecules is really the key to our understanding of life in general, certainly of health and cellular life, even pieces of cells, organelles, different cells from bacteria to eukaryotic cells. And then we won't get into sort of the bigger picture here in this course, but you can also appreciate that, maybe this slide here is, human health centric. And certainly we're going to be talking mostly about data sets and model systems that are giving us information about humans. But these 3 molecules are really available for us to query in any form of life, in any setting, broadly. So you can think of access to the entire web of life and even beyond that into different biospheres. But we're not even going to touch on that. We're going to be very focused and more, let's say, human genome, human proteome centric. And then at the bottom here, I always like to bring in the idea that we're talking about omics, right? We're talking about big data, and we're talking about experiments, both conducted and then archived in databases that are just not possible to access with your human brain and the time you have, sitting down at the lab alone. There has to be a computational infrastructure to that. It's hand in hand with where we are today in omics research, and definitely utilizing that. So that should not be a concept that's lost to you at all, but I like to point that out right away in this lecture. So another little vignette that I like to talk about at this point in the introduction is a term now that, well, when I started teaching this course, I had actually just started doing genome editing in my lab and was part of a project and a project team that we got some of the first taste of genome editing, but for a while I had to explain what that meant to people about genome editing. But now you should all be very familiar with that term. And it has a lot of different meanings. But I think we can all appreciate that our ability to edit a genome or a gene really ties together all this information from DNA, RNA to protein. So that's why I'm putting it here. And the other thing that I've just been having fun keeping track of is when genome editing burst onto the scene and became part of everybody's lexicon was right around 2013. And that, of course, was when we had the first demonstration of editing or changing human genes in a human genome with CRISPR-Cas9. And just watching the fascinating, extremely fast infiltration of this one technique and this simple idea across multiple species, perhaps every species, even bringing the attention of, the United States government and panels of ethicists when suddenly we found and learned that human genome editing experiments were being conducted, and all of the things that unfolded very rapidly after 2013. And then what we were always predicting, and we would always say this, well, you weren't with me, but I would wonder, when are we going to see a Nobel Prize in this area? And lo and behold, late last year, we saw the Nobel Prize awarded. So it's just an interesting way to think about, the big picture of what we're talking about in this course and what can you do with vast amounts of genomic and beyond genomic information. And I think genome editing in the field that we're firmly in now and in the future is a great example of that. So we're going to come now crashing back down to a more focused discussion about proteins and proteomics. Okay. So I like to start with a simple definition, and that's the study of expression, location, modification, interaction, function, blah, 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 of a cell, organelle, tissue, or organism. You should already appreciate that this is very different and more dynamic than other types of omics, because proteins are not localized in one place, and they change extensively from when they're expressed in a cell to where they end up and where they function. So to really get a handle on proteins in a cell or tissue or organism, it's multifaceted. So keep that in mind. Like, we will really break this down into bits of information that we can trace back to individual proteins and then trace that back to the genome. But there are many aspects of this definition that still haven't been addressed by omics techniques. They're only now beginning to be addressed. So just keep that in mind. It's a very complex landscape when we talk about all of the proteins, let's say, in a cell. What does that mean? So we're going to walk through. In the beginning, we have to cover some techniques and technologies. There are a lot of detailed ways that we access a protein, and I want to walk you through some of the basics and certainly still the most dominant techniques in the field. I'll touch upon other techniques that are clearly relevant, but by and large, the proteomics field is still dominated by a single type of instrument. That's a mass spectrometer. So we're going to talk about that. And we're going to talk about what I think is important is like, what is the fundamental piece of data that you're acquiring in a proteomics experiment? And that's important because I want you to understand what are the individual pieces of data? What are the digitized pieces of information that are collected and then brought back into, an omics scaffold or a database or a collection? And then we'll focus on applications. That what I mean by that is how are these technologies and these approaches applied to understanding proteomes and proteins? And I'll use case studies, which are papers from the literature. And then finally, we'll put this all together and talk about how we're continuing to curate this information and putting things into databases. And then we'll also discuss what a pathway is, because we really have to think about proteins working together and connected to each other to really start to understand organization and function. So principles of mass spectrometry, I'm going to try to go through this relatively quickly, but please stop me if you have any questions. So we're going to measure ions, okay? So we're going to talk about ionization very briefly, but very simple. There's this thing we're measuring, and that's a mass to charge. Okay, and we're going to show you what that means. And for this lecture, when I may say proteins, but in the beginning, I'm going to talk to you about peptides. And so what's also important to understand is that for this lecture and for the field that I'm discussing today, we're really measuring peptides, not whole proteins, peptides. So it's the peptide mass to charge that's being measured. But to get there, we have to ionize a peptide. And this is an interesting concept, but it was very critical in the field. So to go after a proteome or to do a typical experiment in proteomics, you need a mass spectrometer. Ultra simple view of it, you have an ionizer, which is your interface. You take a sample, convert it into ions. Those ions are then filtered and detected. Okay, the reality is they're far more complicated than that. And this is an area that I'm just going to sweep to the side. We don't have time to go in to how all these pieces of instrumentations work. Modern mass spectrometers are more like 6 to 10 different types of instruments all packaged into a box, and they work together. Tons of physics, tons of engineering that we're just going to skip. And then, yes, there's 3 principal components to a mass spec, but there's also human. We really haven't been able quite yet to completely remove the human interface from the equation. So there's really an important human operator that's, and here's a picture of a couple of years ago in my lab. There's one of my graduate students working on a mass spec. So with any major breakthrough that changes a field, typically Nobel Prizes are awarded. So in 2002, we saw a Nobel Prize recognizing the ionization technique. Sounding pretty simple, why does ionization deserve a Nobel Prize? It was really, there was a collection of technologies that were the first breakthroughs that allowed us to take a peptide, ionize that peptide, shoot it into a mass spec. And in a nutshell, that was really what brought us into modern proteomics and really gave us access to proteins in a way that we could realize the many, many techniques and examples that I'm going to walk you through. So the ionization, it was a major breakthrough. Mass spectrometers had existed prior to this point, but no one had really been able to make much progress measuring proteins and peptides. So I'm going to leave ionization there. And if you're interested, we can talk about it another time. But again, a tremendous amount of physics, chemistry to go over there, so I skip it. But instead, I want to introduce you to a general workflow. And this is something that still today drives most of the experiments when we start to tackle a proteome. And so there's a sample, and today we're going to talk about proteins, so depicted here. And we'll talk about, different types of samples, right? We're going to talk about an entire cell as a lysate or an intact cell, but also purified proteins. So pull proteins out of a cell and introduce them into a mass spectrometer. To do that, we have to break them up or digest them into peptides. Those peptides are then introduced to a chromatographer. Again, I I'm going to skip over a lot of these details, but just keep in mind that you have an incredibly complex mixture. If you think of a cell, even one cell, loaded with hundreds and thousands of different proteins, and if you convert all those to peptides, we have to have a way to sort of organize and sort that out. And so a lot of that is achieved through chromatography. And then the chromatography slowly introduces peptides into the mass spectrometer, which then ionizes and collects the data. So that's the big picture. And I'll point out little pieces along the way and tell you when certain innovations in the field have really related to different aspects of this workflow. But there's your general workflow. And then what happens in the mass spec, again, I'm trying to boil this down into something that's as simple as possible, but I'm skipping a lot of details here, so bear with me. But depicted here is really the first measurement that's made, and that's a peptide. So here's a piece of data, and I've depicted underneath it a single peptide, because what I'm displaying to you is here are two separate peptides that are resolved inside the mass spec, and they have a characteristic peak to them. So we've detected these ions, and there's two different peptides. And then what's also important is once these are identified in the machine actually physically isolates a single peptide and then brings it over to a different piece or a different part of the mass spectrometer and actually sequences that peptide. And so we're going to talk about what that means. So you have detect a peptide, isolate it, break it up into pieces, and sequence it. So I'm going to walk through this. So the digestion is fairly straightforward. So down here, I've got a reminder that proteins are mostly structured. They're complicated. So you have different levels of structure, and then you can break that down into a tertiary structure, and then you can think of it as a collection of secondary structures. But all that's really out the window. We're not talking about that when we're using this type of mass spectrometry. We're talking about taking the linear sequence of amino acids, that's a polypeptide, and cutting it up. Okay, so taking long polypeptides and cutting it into short peptides using the enzyme trypsin. And it's useful because it cuts very robustly and very reliably at lysine and arginine, which are very common amino acids. And in most proteins, there's a nice regular distribution of lysine and arginine. So in general, you're able to distill a protein mixture down to peptides. Okay. So this is a busy slide, but the whole point is to impress upon you that the mass spectra of peptide mixtures are complex, okay? Let's take a cell, take an entire cell, hundreds and thousands of proteins, all different molecular weights, chop them all up into one big mixture, and you have all those peptides together. And you start to resolve those peptides through chromatography and mass spectrometry. And a component that I want to point out here is time. So we're not taking this entire sample and just doing one shot into the mass spec. We're continuously collecting data. As the peptides come off the chromatographer, they're being injected in the mass spec. And so the entire experiment takes time. And along that, every fraction of a second, all these measurements are being taken. Measure the ion, select the ion, sequence the ion. So detect the peptide, sequence the peptide. So down here, and this is meant to be crowded and complicated. Here is one snapshot, so just a fraction of a second during this type of experiment. And what I've decorated here is a complicated mass spectra, but it's actually comes from just a simple mixture. This is from data from a friend of mine from his lab where, okay, there's 5 proteins in a mixture. And even those 5 proteins can create an incredibly complicated mass spectra. And so I've just labeled in red peptides that are derived from one protein. And then next to those are peptides, labeled here with a little triangle in blue from a different protein. And the point is that if we're simply measuring a mass and a charge, then you can imagine that, well, all these proteins are going to sort out into peptides of defined mass and charged, and that mass is defined by their amino acid sequence. So it gets very, very complicated very, very quickly. And so a simple way we think about it is a mass, okay, is defined by the sequence of the peptide, but the charge is actually defined by that ionization technique that we briefly talked about. But the whole point is that the peptides acquire a charge, and so they create a signal that's based on a very simple distribution of their mass and their charge. And one peptide could actually be resolved in multiple charge states. So that's another layer of complexity. You could have two signals that actually correlate back to the same peptide. But very simply, the peak that we see, this is a peak, right? This signal is on this axis here is a mass to charge window across many thousands of mass to charge elements, right? So a single peptide will have a defined mass to charge. And if you zoom in, and that's what I'm trying to just show you here, is that even in what looks like a single peak, if you zoom in, so a much narrower mass to charge window, you can see there may be multiple peaks there. And what I'm decorating here is that two peptides from completely different proteins could have a very similar mass to charge. Now in this, I've got the laser pointer here over this peak that has two peptides that are really close to each other. By our eyes right now, that looks close. But the way these mass spectrometers are designed, that is in fact not very close at all. They have amazing resolution that can allow them to identify and filter out peptides that even appear that close in mass to charge, but they're easily separated and dealt with by the machine. Okay, so that's a way. What I'm trying to unpack here is how do we deal with all this complexity? Hundreds and thousands of peptides in a mixture, mass to charges that are unique, but could be very close to each other. A lot of this is dealt with in the sophistication of the instrument. And this, again, is to give you a general sense of what does a piece of raw data look like from a mass spectrometer, and what is a mass to charge conversion of a proteome into the primary data of a mass spec. So the second part is the sequencing step. So you'll recall I mentioned that first the mass to charge, or the signal of an individual peptide ion, is collected. Then the instrument chooses that ion to sequence, okay? And the way it does that is it selects it and it breaks it along the backbone. And so the basic principle is shown here. If you had a peptide, and it doesn't matter what the R groups are, so let's just think of this peptide. Let's imagine the amino acid sequences A, B, C, D. Okay. Remember, this is all this machine is doing is measuring the mass and the charge. So we know that a peptide 4 amino acids long is heavier than a peptide that's 3 amino acids long, right? So if you break these 4 amino acids into 2 fragments, one that's 3 and one that's 4, the difference of these two is the mass of an amino acid. Okay, so that if you walk backwards, all I'm doing is, or all the machine is doing, is arranging these peptide ions in decreasing mass, and you know the difference between each is the mass of an amino acid. So this is where the simplicity of peptides comes right into play. For this lecture, there's only 20 amino acids. So that means the distance between all of these fragments is going to boil down to 20 different differences that can all be assigned to an R group of an amino acid. So the sequencing is just that simple. Take an ion, break it up, measure this, what we call a fragment spectra, and you have right in front of you the amino acid sequence. It just happens to be in the language of mass to charge peaks, okay? So I'll show you, I'm gonna bring in a final slide in a moment about, I just broke this down. I said, oh, it's so simple. You can just take an ion and you can sequence it, but there's a big step that I'm leaving out for the moment. So a very important concept that I want to impress upon you right now is about coverage. So you've already had multiple lectures and probably even prior to this course experience with nucleic acid sequence, DNA, RNA. And you know very well that we use next generation sequencing. That's now the current generation sequencing. So for the most part, we can account for every single amino acid, or sorry, nucleic acid in a sample. So it is possible to sequence nearly completely a human genome. If you do an RNA-seq experiment, you're going to account for many times over every single nucleic acid that's in that sample. That is not the case in proteomics. We still don't have complete access to the full amino acid sequence of any organism or any sample. We'll talk about that more as we go along, but here's an example. So if I show you the result of a mass spec or proteomics experiment where we are trying to understand the proteins in this case, it's the membranes of a red blood cell, membrane proteins of a red blood cell. In red are all of the amino acids that I've accounted for in a mass spec experiment. That means I've taken the peptides, sequenced them, and we can see we can see that we've resolved most of the protein, but not everything. So in black are the peptides that we don't see. And there's a long list of why we don't sequence them or even detect them. One of the major realities is it may be a region of the protein that isn't very lysine or arginine rich. That means that it's not accessible to trypsin. And there are other reasons. So one interesting component here, so if you know a little bit about how integral membrane proteins work, you have segments of the protein that spans the membrane, and those tend to be depleted in charge. And so you wouldn't see as many like lysines, arginines, you would see in enrichments of hydrophobic amino acids, okay? And so that's what you're seeing here are the missing segments are sometimes the segments that are passing through the membrane, all right? But then there are also examples of proteins that we sequence quite well. So beta-actin, we get near complete sequence, okay? So the take home is if you see a report of a proteome of any type, keep in mind that typically those proteins are only sequenced a little bit, and we'll go into this in more detail as we go down, the road in this lecture. But the more complicated, the bigger the sample. So if you took like 2 or 3 proteins, you could get a lot more sequence cover. But when you start to do a proteome, you'll get less and less coverage. So there are cases where we can account for, let's say, all of the proteins in a human proteome, but the sequence information boils down to maybe 1 or 2 peptides per protein. That means that the majority of the protein information is not accounted for. We're mapping it back to only a few bits of peptides. And that's super important to keep in mind because it's very, very different than nucleic acid data, where we can really resolve nearly to completion the entire sequence. Okay, so big take home of this, that proteome information is incomplete on the sequence level. Okay, so the part that I glossed over a moment ago, but I'm returning here is, so how does one really interpret a complex proteome experiment? And the answer is you have to rely on computation. Okay, so all of this information is collected. So again, I'm showing you the first scan where these triangles represent intact peptides. And then one at a time, they're picked and they're sequenced. And so for every peak in the first scan, you get a corresponding sequence. And so there are computational steps that put that all back together again. And what's really, really important here is that the only way that this is possible is if we understand the genome of the organism that we're working with. So there's no question that as the genome information became more and more robust, the more genomes we sequenced, the more access we had to the proteomes. And it's really simple. The genome tells you what proteins are possible or what proteins can be found in that sample. So when we interpret the data, we're not assembling the proteins de novo. We're not just taking the mass spec data and figuring out what the peptides are. We're actually matching it. So the search is constrained. We're not saying, oh, this peptide could be anything possible, because that starts to be almost an impossible combination if you take 20 possible amino acids over a large search space. But we know that's not the case, because the order of amino acids in a protein is defined by the genome. So we can look at the open reading frames or the protein coding segments of a genome, and that makes a much smaller database. And it makes then a very straightforward computational step to take raw mass spec data and interpret it back into a peptide sequence. Okay, so the database searching and the computational step is enabled because we have such reliable and accurately mapped genomes. All right. And I may have said something that really quickly, but let me point out, you have to know the genome. I've been in the field for many, many years, and I've never once had just a sample given to me that's like, here, Dr. Reinhardt, guess what this is? No, we always know. I isolated these proteins from a human cell, from a yeast cell, from a Drosophila, and that's your reference genome. You can always plug that into your experiment. You're never having to figure out what the source of the protein is, at least not, I would say 99% of the time. Perhaps you could. It is possible to take, a sample of unknown origin and figure out where it came from, but that's not what's being done in the majority of these experiments.
Dr. Reinhardt, I don't
Yeah.
question.
I hear you. Go ahead.
So let's take the example of cancer where the genome itself is mutated.
Yes.
Right. How would we then start figuring out what proteins have mutated, or sort of what proteins have sort of alternate versions of them available?
So there's really one way to approach that. Well, you could figure that out from the protein data. A little bit more complicated it is possible. So you could find changes at the amino acid level that made your peptide different. But quite frankly, the way it can be done is you use a reference genome. So a genome with a standard amino acid sequence, and then you can also insert a mutation, and you can search your protein data for a reference or a mutant. And that's been done. There's plenty of papers on that where they've shown that you could take a regular cell or normal cell or human cell and a cancer cell, and you could actually pick out and map the mutant protein from only the cancer cell. And again, we know what those mutations are. But it is possible, more difficult, maybe to find a mutation de novo. It's again possible, but a little bit more complicated. Does that answer your question?
Yes. And
again, genome data, huge. You could take the cancer genome atlas, and you could take a collection of cancer cells, and you could pretty quickly filter through it and find those proteins.
That's really cool. I had another question as well,
Yep.
Which was, so we know that the genome is, we know most of the genome, but we also know that it's not quite complete, right?
Yep.
So are we also actively then missing out on proteins when we are just splicing ourselves? So do we completely leave out peptides that are not mapping to the genome?
Yes. And we don't have a lot of time to get into that. There's some details both in the way that the instrumentation works and the limit of the technology and where we are. But most of the genome that we understand has covered extensively the protein coding segments. Some of the missing information in genomic databases tend to be in areas that are not protein rich. But an example of proteins segments that are difficult for genome and proteome are large repeats. So you'll have extensive replicates of, there's proteins that have huge expanses of glutamines, for example, or glutamates. And those are very difficult to sequence and sort out, even next-gen sequencing techniques. And you can imagine that you could produce many protein fragments that would be very difficult to understand. So we do lose some resolution, and there are gaps in the knowledge. So that's absolutely true. But the majority of the information can be map back with high confidence to proteins, but you can lose, like I mentioned before, why do you lose coverage? Why don't I see from the first methionine to the final stop codon? Why don't I see all the information? And some of the reason is for what you pointed out. Okay, so we're going to move along. Great questions. So what I'm pointing out here very briefly, and it will come up again later, I just wanted to introduce the concept. If you're interested, you can read a lot more about it. It's a data acquisition choice. And we've talked about how the first measurement that the mass spec makes is to identify the intact peptides. These peaks represent a peptide. Then the second choice is, okay, I'm going to sequence which ones. So a data-dependent choice is, let's pick the most intense peaks, pick those and sequence them. So if you did that experiment here, you would sequence roughly 10 peptides. But we know that we're going to miss a lot. So let's say all these little peaks down here are also peptides. So if you only pick 10, you're only getting a fraction of the data. So there was a concept of data acquisition that was only recently solved. So now this type of acquisition is a reality, and it's starting to take over the field. So I thought I would introduce it because it is changing the way we do proteomics. And that's, instead of just picking the peptides by the data, we're simply going to take everything. And so these little bars represent that we're not going to just pick this peak. We're going to sequence all of the peaks in this window. And so what we're going to do is march along and sequence. And it's called data independent because we don't care about how much signal there is. We're just going to sequence everything. Okay. So this type of workflow actually is what's going to bring us into a deeper coverage, more reliable and robust proteomics in the future. There's still a lot of work to be done, but it's starting to take over the field. So I thought I would introduce it here and give you some references for further reading. Okay, so we're going to leave that. Here's another data-rich slide. And all I want to point out is the following. At the top, I wrote, the pace of proteomics is set by a combination of techniques and technologies. That's been true for 20 years of mass spectrometry. The more we advance the mass spectrometry technology, the better and faster we're getting at proteomics. And that's illustrated here. I've got 2 arrows and 2 references. So I put a blue arrow here about a paper in 2001, which is really an important paper. It was really what was the first proteome that was published. So this group published what was the yeast proteome, okay, using a whole bunch of different techniques. But that was in 2001. And they were using some of the earliest type of technology that allowed you to make these measurements and deduce all of the proteins in a cell. It took them roughly 82 hours just to do one yeast proteome, okay? There's a lot of details I'm skipping over, but the point is this. In 2013, so that's 82 hours, they got about 1,000 proteins, let's say 1,500 proteins. Okay, yeast has 6,000 proteins that we know of, okay? So that's not a whole proteome right away, but it was a major achievement. It was what we thought was like a real proteome experiment. But by the time we get to 2014, the speed and depth of peptide analysis, driven just by the mass spectrometers themselves, brought us to 1 hour of acquisition time, got us almost 4,000 proteins. And it turns out that one yeast cell is pretty much only using 4,000 or so proteins, maybe give or take 50 or 100. We don't think a yeast cell actually uses all 6,000 of its genes at any point. So a proteome from a yeast cell that's about 4,000 proteins is pretty much the entire protein content of that cell. So what I'm telling you here is, think about if you had to do an experiment and it takes you 82 hours, but now you have technology where you can do that same experiment in 1 hour. That's really going to change the type of experiments you can do and the power of proteome technique in general. So that's 1 decade jump, give or take. And that was a huge boost for the field. And that really is what started, by 2014, we started doing some of the more ambitious experiments that we're going to talk about. So proteomics is all about the sequencing technology and the speed and the efficiency of the mass spec. And that's it in a single graph. Okay, but here's some facts. There's still major challenges. So proteomics is sample limited. All right. So if you're sequencing DNA or RNA, most of the time you're using a polymerase. And why is that important? Because with a polymerase, you can make an exact copy of that genomic information or the transcriptomic information. You can amplify it. So you take what the cell gives you and you amplify it. You make a lot more of it. And that gives you more access. Okay, you can't do that with proteins. So I wrote here, proteomics has no polymerase. You can't take the proteome of a cell and amplify it like you would with PCR or even reverse RT-PCR. That doesn't exist for proteins. So you're stuck with what the cell gives you. Okay, and we'll talk about areas where that is really still limiting the field. That you have to deal with what the cell gives you, and so you're sample limited. That is still a major barrier to proteomics. At the end, when there's time, I'll tell you about single cell proteomics, and we're very far away from that, but it's beginning to come online. No polymerase, that's huge. And the mass spectrometry itself has a limitation. We don't yet have mass spectrometers that can provide amino acid resolution of a proteome. And that's the point I made earlier. You can, let's say for argument's sake, you can sequence every base pair of a genome. You're not even close to doing that in a mass spectrometer. You're always going to miss whole proteins, whole stretches of polypeptides. Many peptides are not going to be in your data set. Always going to have that limitation for a good long time. And really until these two major roadblocks are solved, we're really not going to have sort of the next gen sequencing for proteomics, to put it simply. We're still behind the nucleic acid field by probably order of magnitude or more because of these two major problems in proteomics. And groups are working on it. I'm excited for the day where they're solved. I think they will be, but we are where we are right now. Okay, so I'm going to wake you up a little bit. So I'm kicking this back to you, so pay attention. Here's a challenge question, okay? So I expect to hear an answer. Okay, so we just talked about mass specs and limitations. So here's the idea. I have a simple proteome. Here's a cell. It has 4 proteins, okay? And so I take and digest all those proteins up into a sample. There they are. So there's a green one, there's a blue one, okay? Now, I happen to have the world's worst mass spectrometer, okay? It can only identify one peptide, all right? How can I analyze this proteome? In one experiment, I can only tell you, I only can find one peptide, okay? And there's 4 proteins. So how can I map or sequence this proteome? Anybody have an idea? Don't be afraid.
You can do multiple experiments though, right?
Yes.
Once you have identified protein 1, you could probably use some digestion enzyme that specifically targets protein 1.
Good idea. Pretty close. So, okay. So we can do one protein at a time, one peptide, and the mass spec's awful. So what if I could do multiple experiments? What might be another way to increase the coverage of this proteome?
Knock out the gene that codes for protein 1, and then like sort of.
Okay. All right. That's a cool idea. Probably not going to work. It's definitely not going to work if you think about doing that experiment for what we're going to talk about next. But I'll give you one answer. Okay. It's a great one. And you could get a long way and do some cool work with that, what you mentioned. But what I'm driving at is there's some very simple techniques that we can actually do before we use the mass spec. So you're almost there in your answer. So one could be chromatography. And in a way, we do this already when we separate out peptides. So if you could separate the peptides into groups, you could have a fraction that only had protein 1, you could identify it. Another fraction, peptide 2, identify that 3, 4. this is actually how we dig deeper and deeper into proteomes. We can separate those peptides away from each other and make individual measurements. So the concept is fractionation. Fractionate them out, separate them from each other. Extremely powerful way that we can process a sample and dig deeper in the proteome. So this is, if you can think about it, this is another type of fractionation, but it's also a very powerful way that we can go after a proteome. What if you had an antibody for each cell, each protein in the cell? So that's what this picture is here. You've got this antibody. I've got an antibody for protein 1, 2, 3, 4. And then I take and I open up that cell and I throw in an antibody and I pull out protein 1 and I digest it, shoot it on the mass spec. Then I do the same thing with 2, 3, 4. And there you go. I've sequenced all the proteins in this proteome because I've had an antibody that does that. Okay. So the concept here is, even with the limitations of the mass spec, we're really, really clever. And we've thought about many ways that we can actually, deal with the proteome and all of its complexity and use the power of mass spectrometry and start to get all that information. So you'll see these two themes over and over and over again in the coming examples. But as prepare yourself, I don't know of a cell that only has 4 proteins. We're going to be talking about cells like yeast that has 6,000, or humans that at least have 30,000, but probably more or closer to a couple million proteins per cell. So it's going to get a little bit more complicated. Okay, another slide full of words. This is a resource slide. The concept here is I wanted to take a model organism, yeast, and show you the progression of proteomics in a decade. Okay, so this happens, it's starting to get old. I mean, 2000, like 20 years ago. It's still a benchmark, multiple benchmarks in the field. And what this is breaking down how did the field get from proteins to proteomes, right? Because that's what we're talking about. We're talking about omics technology that allows us to understand, measure, and do experiments with all the proteins in a cell. And that information goes into a database, and you can leverage that along with organism's genome. How did we get there? Okay, so this is sort of the 1 decade tour of how we took yeast and started with the proteome at low res all the way to high res, okay? So I'm just going to breeze through a couple of these examples, but again, each of these papers is referenced on that slide. So a technique that I want to mention just briefly is that there were ways to do a proteome-wide experiment without a mass spectrometer. And this is a classic technique called yeast-2 hybrid. And there's a lot of words in this cartoon, but this is all I'm trying to impress upon you. This technique is pretty simple. You take the genome and you break it up into pieces, and then you make gene fusions, okay? So one gene fusion has a piece of a transcription factor, and the other gene fusion has another piece of the transcription factor. So what happens here is if you've got 2 pieces that interact with each other, you turn on the transcription factor, and in this case, the yeast cell is going to live on a selectable media. What does that mean? That means that you can, and this cartoon shows bait prey. What they did is they took all of the yeast genes and made a whole collection of bait and a whole collection of prey. And then the experiment was mix them all together and ask the questions, which 2 reconstitute a transcription factor activity that gives you a cell, okay? And what you got was, what I didn't show here, is in these 2 papers, they were able to deduce the protein-protein interaction networks of the entire yeast genome, and therefore the entire yeast proteome. Okay, it was a very powerful defining moment in the field because this was one of the first proteome-wide experiments. Okay, so it's worth mentioning because that's what really started kicking off the field. It's possible to do a proteome level experiment, and then a few years later, a group decided to use the same technique, and instead they used roughly 7,000 human genes, and they did the same experiment. They asked the question, 7,000 genes from human, how do they interact with each other? How do the proteins interact? Using yeast as a chassis. And here they produced the first high content human interactome. So this hairball represents all of the different combinations that these 7,000 genes produced in this experiment. And they realized that 7,000 genes gave rise to more than 10,000 interactions, implying that protein-protein interactions are very, very abundant and really dictate the organization of proteins and proteomes. So proteome level experiments without a mass spectrometer. And they map some disease genes, but for time, I'm gonna go through this. So just to introduce a concept that maybe you've seen before, but let's just go over it. When we're drawing these networks, okay, of a protein-protein interaction, you have a node and an edge. The nodes, and what we're going to talk about, are different proteins, and the edge defines the interaction, right? So you'll see cool networks like this, where a protein complex is shown by how they all interact with each other. And then people constantly cull these databases and try to deduce how many protein interactions do we know of in human cells. So at 2010, this was 100,000 interactions that were deduced from databases. And you'll see that we've gone way beyond that now in 2020. And so protein-protein interactions are important. This is how proteomes are organized. So you can have physical interactions, indirect. So some examples are how enzymes might collaborate in a metabolic process, kinase-substrate relationships. So there's a lot of information in protein-protein interactions, okay? And the reason I'm going into detail is, besides just the sequencing a proteome and reporting these cells make these proteins, the next most abundant database information we have on proteomes is on interactions. We now have very, very rich data sets across multiple organisms of how proteins come together and work. So it's still one of the most rich proteome level databases that exists today. Okay, so I'm just gonna briefly touch on a technique that was developed for yeast, still being used today, but it's just that concept of, if you want to start pulling out protein complexes, how much you do that. And so one concept is to use an affinity. So an affinity tag, and in this picture is actually a dual affinity tag. So there's the protein of interest. And the question is, in a cell, what proteins interact with that bait protein, okay? So you attach a tag. In this case, you're using two different, one is you can pull down with an antibody interaction, and the other one you can pull down with like a calcium-dependent interaction. The details are important. What I want you to just think about is this. When you do multiple steps of purification, what you're eliminating are these white circles, or these proteins that aren't part of the complex. So you're starting to purify out just the proteins that are touching each other, okay? Then you can take that protein, you could shoot it in a mass spec, and you can ask the question, what proteins are in this complex? So the reason I'm walking you through this is because what happened was a group decided that, wow, this is really useful. Why don't we go ahead and tag every single yeast gene? So they made almost 6,000 different strains of yeast, all that contained the same tag. So then what you had was 6,000 different strains, each had a common handle, and you could just go ahead and pull out all of the protein complexes from yeast. And that's in fact what was done. There was a consortium of labs that got together and performed many thousands of purifications, and they did many thousands of mass spec runs to deduce how the yeast proteome was organized into protein complexes. And so that data is distilled here. It's still a classic paper. Nevin Krogan, who now has his own lab in California, you may have heard of this guy. His lab actually mapped the COVID proteome, which came out in rapidly after the start of the pandemic. So he's still very much in the proteome interaction game, and he's still one of the leaders of the field. But what we saw here was the first time an entire organism organized into protein complexes. And this was a major achievement in the field. It was really the first time that we did this, and it had far greater resolution than that first yeast-2 hybrid experiment, okay? But it was a massive undertaking. And, you know, it's sort of like the Human Genome Project sort of in protein space. So it kind of has that type of landmark to it. So the reason I spent some time with yeast is to just give you these examples so you could really understand, how did the technology and the cleverness of the labs sort of get us to the point where we could do a proteome experiment. But the question is, like, how do we get from a simple single cell organism with only 6,000 proteins to humans? Okay. And there's a couple answers, but one of the simple ones is, well, we can try to break it down. So early on, the goal wasn't to do the entire human cell, but let's do biological classes, or let's do networks of common proteins. So this is one of those landmark studies that came out, like 3 or 4 years after the yeast interactome. This group collaborated to just understand how autophagy was organized. And so what they did is very simple. It's, again, a very busy slide. I'm overwhelming you with information, but the simple take home is very similar to what they did with yeast, right? So in the yeast experiment, you tag all the proteins and you do, 6,000 purifications. You have 6,000 complexes. You run 6,000 runs on your mass spec. Here they're like, okay, we'll do 65 proteins that we know all participate in autophagy. And so they did IP, so immunoprecipitation. So they had an antibody that pulled out only 65 baits. But what they found was, only with 65 baits, they reassembled complexes that totaled almost 3,000 proteins. So the lesson was, as the genome scaled and the proteome scaled, of course the interaction network scaled. So we knew what we were in for when we were going after this type of question in humans: a lot more protein complexes. So this illustrated the problem, right? With only 65 of the 30,000 genes available, you're pulling out almost 3,000 proteins. And then inside those 3,000 proteins is a huge network of interactions, right? So this was the defining moment. This is what we want to understand. And I threw this slide in here because this type of work is being done today, where interesting sub-fractions of the human proteome complexes are being investigated by multiple groups. And this is just an interesting group of papers because this happened to be a signaling pathway that was interrogated. And 3 different labs did this experiment. And it turns out that they got very different answers. So it's just a pause in all of this information to point out that this is a very technology heavy field. This is a very methods driven field. And there's still a lot of imperfections in it. So you have a lot of experimental variability that's still being wrestled with as a community. So this kind of highlights that. So there really isn't yet sort of the ground truth proven set of protein-protein interactions that defines a human cell. It's still a work in progress, and there's a lot of experimental variability. So just keep that in mind. But nevertheless, we've made tremendous progress. And here's the example. When I was first starting teaching this course, it was exciting to talk about yeast. It still is, but I thought, okay, clearly we're going to start doing this with the human cell. Someone is going to go ahead and define the interactome of humans. So it turned out to be the same group that did that autophagy group. It's led by Wade Harper and Steve Gigi at Harvard. So what they did is they used the same similar approach to the yeast project. Insert a common tag, so a little protein handle on every single human gene. Put them one by one into a cell culture, express it, open up the cell, pull out that complex. That's what this little picture is here. The antibody grabbing the tagged protein, shoot it in the mass spec. The difference is, instead of 6,000, they're going to have to deal with 30,000. And so what has resulted is an ongoing effort to map the entire human interactome. It's called the bioplex. The first paper came out in 2015, where I think these are the stats up here from the first paper. They did close to 6,000 IP mass spec experiments. So that's almost the same amount as they did for yeast, but they identified over 50,000 interactions. Okay, so we're starting to see a scale. And of those 50,000, they contained about 10,000 different proteins. So they only had to pull down 5,000 proteins and they identified 10,000. So that's what's sort of exciting about this is that you don't have to have full coverage of the proteome or the genome tagged. You're going to still get the information for the complete proteome. So in other words, we don't have to saturate genetically the human genome to really start to understand how all of the proteins could be accounted for. And just last year on bioRxiv is now Bioplex 3. Bioplex 2 is in a Nature paper, Bioplex 3. And we always joke, like, probably it'll be Bioplex 4, 5, 6, 7, 8, 9, 10, who knows? But we're starting to get to that point where we've almost defined all of the proteins, and at least have a handle on how each protein can interact with others. And so we're towards this map. But the take home I wanted to point out here is, lo and behold, proteomes are organized into complexes. So I have side by side here the Krogan paper from yeast and the Hutland paper, Bioplex 1, and we see a lot of conservation. Okay, of course there's differences. Humans going to have a different complex organization than yeast, but there's a lot of core complexes that are the same. And in principle of how do you design a cell out of 30,000 proteins, you design it by organizing those into complexes that collaborate to perform all the different biology that we know about. So it's really exciting to see that you have conservation of the organization of the proteome. And that was only possible by doing these very, very resource heavy and long-term projects to distill the proteome. Okay, so a little bit about the field. So this is just tracking protein-protein interactions that have been reported and deposited. And this is everything. So this data set is mostly human, but there's a lot of other interactions there too. So I'm sort of lumping it all together. And this is still one of the central hubs for curating this type of data. It's the INACT database hosted by EBL. And so what I'm showing you here is, since 2011, how this database has been growing. There's a couple inflection points. This one here is Bioplex. And so a lot of this information is coming from the Bioplex project. But notice that, we're not identifying new proteins. And that should be obvious, right? We know what the proteins are. We figured that out when we sequenced the genomes. So that part of the database doesn't grow too much. Yes, you may gain new access to a protein, and it may be part of an experiment that wasn't around in 2011. So it does grow because there's more and more access to those proteins. But what grows is how they all come together, right? That's what's had this tremendous growth. And in last year, we crossed the 1 million interaction point. So, again, this is the most robust and complete proteomic database that we have. So it's very useful, and it's still growing. It's still very, very active in the community. And I always just plot the growth. And for the first time I really saw it lag. It was usually each year it got bigger and bigger, but the change from 2020 to 2021, we fell off a little bit. Maybe Bioplex is reaching saturation, or maybe that's the pandemic. Time will tell. But compare the million interactions in a decade ago, we only had 100,000. So the growth of the human interactome has been tremendous, and an order of magnitude. Okay, so we're going to start talking a little bit about quantitative proteomics, and we're going to hit our stopping point at 2:15, and then we're going to pick this up at the next lecture. But before I get into that, any questions from any of the material to this point?
Can I ask?
Know the order?
Yeah.
Okay. 2 answers. 1, from the genome. Okay. So most genomes that have been sequenced have been annotated, and the open reading frame, or the coding segments of that genetic information, have been defined. So you can go to a database, you can identify the coding sequence that codes for proteins, and the very first amino acid is always methionine, and then it's set up from there. And we know where proteins begin, and we know where they stop. So when you start a proteomics experiment, you always know the proteins that are being generated by that genome, and you know the order of amino acids, because that's what the gene encodes. The gene always encodes start to finish, N terminus to C terminus, the polypeptide. When we then measure in a mass spectrometer, we take an intact peptide, let's say it's simplicity, it's 3 amino acids long. The mass spec isolates those and it breaks it into 3 pieces. Each piece is 1 amino acid difference. And the difference in mass between each of those fragments is the mass of an amino acid. So the difference between 2 fragments, let's say it's a methionine. Methionine is always going to weigh as much as a methionine weighs, okay? So we know by the mass of the fragments which amino acid has been removed. And when we know that information, we can map it to the known protein coding segments. Again, you have to know the genome, and you have to know the way the proteins are defined by that genome, and you get your sequence, and you're just matching. You're like, oh, I've got a methionine, an alanine, and a glycine. Where does that map to the human genome? And you can position that. And so you know that you've identified that peptide. Now, there are ambiguities. It's entirely possible to have the same amino acid sequence from 2 different proteins. That can happen. And so if that was the one piece of evidence, you had one peptide, it was the same, you wouldn't know. Are you measuring, which protein are you measuring? But what we do frequently get is we get 5, 10, 15, 100 different peptides. Those all map back to a single protein. And so once you get increasing coverage, you get increasing confidence that you've assigned the peptide sequence to the actual protein of origin. Okay, that was a longer answer than you were expecting, but it's a genome combined with the mass measurements we make of the order of amino acids from that peptide sequence. Okay, great question. All right.
Thank you.
Yep. So let's. No,
Oh, okay.
we'll talk about that. We're going to get into quantitation. Okay.
Okay.
So perfect transition. All right. So I like to set it up with this concept of, all right, so we've talked about just take a protein sample and sequence it. All right. And you're going to get a parts list. And you will know which proteins are contained in your sample, but that's usually not enough to understand function. Okay. So what we want to do is try to understand the function. And really, if this clock example, if I just like broke a clock up into pieces and you could identify them, and you're like, oh, I have a lot of gears and stuff. But if you started to learn how they were put together and you assembled from those pieces a clock, you might be able to understand the function. Okay. So the aim of quantitative proteomics has really been to start to try to assemble a functional story behind that. Okay. And it's important. And that's why I illustrate this, because it's not enough just to know what proteins are there. We want to start to understand how do they fit together and how do they work. And so if we can quantify and compare different proteomic states, we can also start to understand function. So like this clock, if I made 3 different measurements 3 different times and I could quantify the difference, I could start to see that the hands are moving on the clock. Like, oh, this is telling time. Okay. So that's the concept, right? We're trying to understand function by quantifying and comparing. So really what it comes down to is comparison. What's the difference between this clock and that clock? And what is the confidence of that difference? Okay. So just reminding you of the workflow again, we went over this, but you're going to have a sample and you're going to rip it apart into peptides and you're going to measure those peptides and sequence those peptides. Okay, but here's the big problem that we face right away. And this will probably be the last concept of the day, that it's the data, the mass spec data itself, the primary data is not quantitative. Okay. These signals do not tell you anything about the abundance, and you can't compare one to the other because the ion intensities, it just doesn't work like that. Again, unfortunately, I have to skip over a lot of this information, but I will just tell you this. this signal doesn't tell me how much peptide is there. It doesn't. So, and this is a great example. You might look at this and be like, oh, that peptide is the same amount as that peptide. No, that's not the case. You can't just take that signal and say, oh, there's 2 moles of each, we're done. No. So the signal you get back from an experiment does not give you quantitative information. But we can get around that. And so what we're going to talk about is ways that we get around that to quantify a proteome. And so here's an interesting example. All right, so I can't quantify the abundance of proteins in a sample with just the mass spec intensity, just the ionization, okay? So here's a red blood cell membrane proteome, and this is a gel. If you haven't run a gel, I feel bad for you, but most of you probably have run a gel or seen a gel. So what you do is you take the proteome, you run it in SDS page, and after it's done, you stain it blue. So here you have the proteome of a red blood cell. And this is a big protein at the top. It turns out this protein is like 80% of the proteome of a red blood cell. Spectrums, these 2 spectrums are huge proteins. And then all of these proteins arrange themselves in size. So as you walk down the gel, you see bigger, more abundant proteins, and then you see smaller proteins that are moderately abundant. So right here, I'm getting a sense for how much spectrin versus how much actin is in a proteome, okay? Very simple quantitative proteome experiment. I know there's more spectrin than actin, okay? You can take, I just said, okay, I called this 16. I understand now 16 proteins in this proteome. Take the same sample and I put it into a mass spec. And I just sequence it. Same workflow we talked about. And then what I do is I count the number of peptides and I count the number of times I've seen that peptide. So the number of observations. And it turns out that in general, the more abundant proteins produce more data. You get more peptides, you get them more frequently. It's just literally by mass, by abundance, okay? So what I did in this experiment is I took the mass spec data, assigned it all to the proteins, and then just lined it up, the most data to the least data. And what I'm showing you is that the most of the data comes back to spectrin, which is the most abundant protein in the sample. The next one is this thing called band 3, and then band 4, and then down here, less abundant than spectrin is actin. And so it turns out that a very simple technique that's used all the time is what we call spectral counting. You just count the number of mass spectra and curate that. And that's what I've actually put here is the number of unique spectra that I counted for each of these proteins. And you can start to get a sense of the quantity that's in that complex. And so that is it's still used today very often. And in fact, that principle is what was used to define the yeast interactome and is what is used to define the bioplex human interactome. You do the pull down of your protein complex and you use spectral counts to answer the question, how much of each protein in complex is together, to get a sense of the quantity and to get a better sense. And what we're after in a simple experiment like that is you anticipate that a real protein complex, the most abundant proteins in that sample are in complex with each other. And things that are not truly in complex, that might be background, are far less abundant. So you can count the spectra and you can get a much higher confidence of the proteins in complex by simply applying this very simple quantitative technique of spectral counting. All right, so that's 2:15 exactly. So we're going to stop here. And so we'll pick up with this slide next time on ways that we quantify mass spec information that gives us much more access to biological questions. So feel free to jump off now. I'll see you next time. I'll stay on for a couple minutes if you have any questions. And if you don't, then I'll check out too. So thanks for your attention.